Welcome everyone for today Hematology the CME, which is organized with the help of ImmunoACT, which is the first Indian company which is they are manufacturing the CAR T cell, as well as Dr. Joydeep. Now he is a very well-known name in Myanmar hematology. So welcome Dr. Joydeep, and thank you very much for Professor AAG, Professor Senwin and others doctors who help us to organize this today's CME. So we will going to have a 3 lecture. First from India, from ImmunoACT doctors, and their honor the person who started this company. And last lecture will be with Dr. Joydeep Chakrabartty. And so I will request everybody, please ask all your questions possible. Like when I was talking to Dr. Senwin and Dr. Joydeep, I asked, in your hematology, there are a lot of gray area. So some doctor do something, some doctor do something. So they both say, because it's a new subject, so not any reference is there. So we try our best. So first of all, thank you very much for taking out your time. And another thing you must be seeing, we are arranging that full video recording of this CME. And after that we will upload on our 4-Hour Health Talk YouTube page. So anytime you can go through for your reference. And Dr. Joydeep is always available to discuss and give advice for your any difficult case. And with the help of Professor AG, now he's coming every 45 days in Myanmar. So if any patient you think is complicated, you need a second opinion, then they can visit to Victoria Hospital, or when Dr. Joydeep is not in Yangon, we can do teleconsultation also, because as per the Myanmar Medical Council, because he is registered already, so we can legally do teleconsultation. Like if doctor is with Myanmar Medical, can do even teleconsultation also, but that is also not legal. So with this, Mr. Says, I will request you can start your presentation. Now floor is yours. Mr. Says? Yeah.
Please. Yes, can you hear me?
Yes, we can.
Yes, so Mr. Ravindra, Joydeep, and all the dignitaries, thank you for coming together on a Sunday morning. I know it is a little bit of a work to get everybody together on a Sunday morning, and also apologies that we could not travel in person this time, but we will in the next opportunity. We will definitely also travel in person, but we have some urgent work over here. So with that, just to set the context, my name is Shirish Arya. I am co-founder and director commercial here at ImmunoACT. And along with me, I have a panel of colleagues, each specializing in different aspects. So I will start with the introductions. First and foremost, there is Dr. Kunal Goyal. Kunal is a part of our medical advisory group. He also consults at the Kokilaben hospital here in Mumbai, and works very closely in many of our cases and data management and patient management. Dr. Bindu, who was senior medical advisor based in Chennai, has been with the organization now for more than a year. There is Dr. Ridhi. She's also a medical advisor based in Mumbai. And then we have Dr. Vivek. Dr. Vivek is part of our international collaborations team, and Karan is also part of our international collaboration team. So Karan and Dr. Vivek, they basically lead all our efforts as far as international collaboration is concerned. So we are all here, and some of us will of course travel down as and when we get the next opportunity. So with that, basically, what we will do is we will just spend another 15 minutes giving a very quick summary of what ImmunoACT is, which is the company, and the key aspects of CAR-T management, what are the clinical data looks like, and what is the key aspects of toxicity management that needs to be taken care of, or not just toxicity management, but what are the different aspects of care prior to CAR-T, prior to infusion, post-infusion. It's a difficult topic to cover in 20 minutes, but we'll try to crash it in. Then Dr. Joydeep, of course, will take you through some of his data points, and he has a very vast experience, as all of you know. And then maybe we can just do a wrap up and try to understand how we can help the patients who are from Myanmar, and how we can make it both as a company and from Dr. Joydeep's clinic, both in Myanmar and this clinic in Kolkata, how we can support you to the best of our ability. So that's the agenda today. So Vivek, over to you.
Thank you, Shirish. Thank you, everyone. Good morning, all the eminent doctors and state company that has a fully integrated platform of CAR-T cell therapy in terms of its R&D, designing, capability. As well as manufacturing at commercial scale. We were established in 2018, and the seeds of our establishment were actually sown in an institute called the Indian Institute of Technology, Bombay, which is an eminent institute for engineering, but also has a very prominent life sciences division. We are headquartered in our CGMP facility is based in Navi, Mumbai, India. And we have almost now 200 plus employees in the past 2 years since commercialization that we have achieved the market authorization of the product. Our vision is to become a global leader in cell and gene therapy through our translational research, and the mission remains to ensure equitable access to innovative cell therapies for every patient who may benefit. Next slide, please. I would like to introduce all our executive and senior leadership team. Helping the entire setup is Dr. Rahul Goyal, who is the founder and chairman of the ImmunoACT. He was instrumental in bringing the CAR-T innovation to India by initiating the research activities back in 2013. That's when he moved from US after his postdoctoral research work along and working on companies at Harvard as well. Dr. Athar Arulkar is our co-founder. He is the scientific head. Shirish, our co-founder and director for corporate strategy and business development, whom we were speaking earlier. Rajat and myself. So Rajat is our CFO, myself working in global collaboration. And then we have eminent scientists working with us in our R&D divisions as well. Next slide, please. I would like to bring you a notice and pause a bit here, because we have the who's and who of the world in the CAR-T space as our scientific advisory and board member. So Dr. Carl June, who was a pioneer in initiating the CAR-T therapy in the world, is our key scientific advisor. Dr. Avery Posey, Dr. Satwan Ilaku, who was instrumental in bringing the CAR-T therapy in lymphoma, and is currently actively involved in many ongoing research and development activities of next generation products. Dr. Gaurav Narula, who was instrumental in our clinical trial and also have now been the PI for our pediatric clinical trial. Dr. Nirali Shah, based in NIH, again a very key advisor in all our processes since the inception, and also as and when we evolve our pipeline for future products. We have Dr. Ravindra Nath and Dr. Satya Narayan Sharma, who are our board members and also our strategic advisors. Next slide, please. Yeah. So I would just like to walk you through the timeline in terms of how the ImmunoACT evolved. So as I said, back in 2013, when Dr. Rahul had this idea of having this innovative CAR-T therapy being part of Indian ecosystem, and where the R&D activities were initiated. These were the plasmids, the electrical vector system that is necessary for CAR T manufacturing were all indigenously developed and designed at IIT Mumbai. In 2020, an proposal for an IND was submitted to the National Regulatory Authority of India, called CDSCO, that comes within the umbrella of Ministry of Health, Human and Welfare, of the Government of India. And a clinical trial began at a very strategic institute based in India called Sedona Memorial Hospital. It is a key seat of cancer treatment, not just in India but throughout Asia, which oversees many patients in an annual basis, not just for hematology but for even for solid tumors, etc. Our phase 1 first in human dose was administered in 2021, and within 2 years we were able to progress our trial from phase 1 dose escalation to dose determining to phase 2, where the efficacy and safety were also evaluated. And we got our, eventually, based on the successful clinical readouts that we reported for the product, we received the approval in October 2023 and market authorization for Kokilaben autoleucel, now called as NextStar19. Since then, in the last 2 years, and in fact tomorrow will be our 2-year anniversary since the product got approved, we have been moving at a pace to ensure that the access to the NextStar19 is immensely reached to almost all the patients who represent the highest unmet need in our country. We have been able to onboard more than 90 plus premium hospitals across the length and breadth of the country. We have successfully commercially infused more than 400 patients in the commercial setting. Beyond this, we have also been very active in publication, in our dissemination of the information, our learnings through publications, where we have published our real-world data, our clinical data from phase 1 and phase 2 studies, as mentioned in the Lancet Haematology, which is one of the key journals in the medical fraternity. We have been part of many conferences, international, like ASH, EHA, the European Hematology Association, American Society of Hematology, and ICT. And in last June this year, we have, in June 2025, we have also received the patent from the US Patent Office for the composition of matter patent for the NextStar19 as well as for the manufacturing process. Next slide, please. So with that, I would like to transition to the product. NextStar19 is the world's first humanized second-generation anti-CD19 CAR T. The fact that it has a 4-1BB co-stimulatory domain provides a very long persistent profile to the product, which is relevant for its durability and durable response. As I mentioned, the phase 1 and phase 2 studies were conducted in India, based on which we have received our market authorization to the regulator. And the approved indications for this product are patients which are greater than or equal to 15 years old in relapsed refractory B-cell lymphoma or relapsed refractory B-cell acute lymphoblastic leukemia in India. The product is infused as an IV infusion in a 100 ml bag that is supplied as a product at the center, at the infusion center, and the manufacturing is now robustly established, and we have a very high manufacturing success rate. The product shelf life remains 3 months from the manufacturing in a cryopreserved self-suspension state. Next slide, please. Yeah, this is, I would like to have your attention for this slide for a moment, because this would set the basis of the discussion further. But it's sufficient to say that the robust vein-to-vein cycle is being now very well established by our manufacturing team. And this representation depicts the systematic time of how an infusion to the patient occurs and how an apheresis is being adopted for the patient. So, to begin with, the patient first counsel and a selection of the patient happens with close consultation with the doctor. Once a patient is identified who can be infused, and I'm sure Dr. Kunal will provide further clarity on how the process begins and infused. The patient, the sample of the patient, the leukapheresis starting material is then collected at the hospital site and then shipped across, at least in India, at 2 to 8 degrees centigrade by our logistic third-party partner. And then we have the initiation of our manufacturing process. So the T cell activation and selection are done at the site in a CGMP facility, which then subsequently leads to lentiviral transduction. There's expansion of the cell product, the in-process QCs and the release QCs that are relevant and are important before the product is sent for formulation and dry preservation. Once the quality assurance is complete, it is again then shipped back at -80 degree drivers of state at the site of infusion, where the patient receives the infusion in an ice. Next slide, please. I would like to highlight few key differential capabilities of the Next-19 as a product. So, in our manufacturing optimization process, we have been able to achieve a very high rate of transaction efficiency, which is almost as per the standards of global standards. The dose, as I said earlier, is 526 cells per kg, and we, in order to have a successful manufacturing carryout, require 210:9 ALC count prior to the manufacturing. So that's a prerequisite. Our manufacturing success rate is industry leading, wherein we represent around almost 99% of our batches are successfully manufactured. Another key highlight for the product is the manufacturing time, the vein-to-vein time, or the manufacturing time remains at around 16 days, and the vein-to-vein time is 20 days, wherein from the apheresis to back to the infusion of the patient. Even from the perspective of post-infusion in India, at least in the trial setting, the average post-infusion hospitalization remains at 10 days, which actually speaks about the safety profile, a world-class leading safety profile that the product has. Next slide, please. So, to give you context into the overall product as well as the company's strength, so ImmunoACT's advantage broadly are based on the fact that the plasmid, the lentiviral vector system has been indigenously developed that provides us an optimized manufacturing batch planning capability where we are not constrained with the supply of the material, key material for the packaging manufacturing. We have established very high efficient transaction rates and expression rates of the party that is also instrumental in driving the cost economics. And over the last 2 years, we have successfully demonstrated robust commercial abilities to provide the manufacturing, provide the product to the patients in a timely manner. And overall, with all the indigenized platform that we have developed, it has led to a significant reduction in the cost of the product as compared to the parties that are available in West. In fact, it is 90% reduced in its product price compared to the other anti-party products available in the Western world. So with that, I think I will request Dr. Kunal to proceed to the subsequent slides, the clinical results. Thank you.
I think, Dr. Vivek, thanks a lot. And before Dr. Kunal starts, I think I just wanted to underline, if I can say the gist of what Dr. Vivek was trying to say. Here we have an opportunity, and something that we are really proud of is we have an opportunity where we have brought a world-class product at a price which is affordable, significantly affordable to anywhere in the world. I think, and the validation now that is done with over 500 patients, so both their clinical results the manufacturing capability, the scaled up manufacturing capability, all of that basically we have brought it together. So I think I just wanted to highlight that the robustness of the platform and the product before we get into the next phase. Thank you.
Hi, good morning everyone. Thank you for sparing Sunday morning. And I will be highlighting about the clinical trial aspects as well as the efficacy and safety part, along with touching some of the part of toxicity management as well. I believe Dr. Joydeep Chakrabartty sir will be continuing with all those toxicity part as well and the clinical cases later on. So going forward, in the phase 1 and phase 2 clinical trials which was conducted primarily in Tata Hospital, Mumbai, and if you see the results of phase 1 and phase 2 trials, before enrollment, can we go to the next slide? The patient's characteristics were who were selected had a very bad biology, and for BNHL, the average age was around 51 years, and for ALL, they present the younger cohorts, so they have an age of 22 years. Majority of them were male, and most of the patients in NHL have received more than 2 lines of therapies. In BAL, most of the patients were primary refractory patients. The performance status, as in the trial, it was kept less than 2. If you see, the primary refractory patients were representing 1/3 of the cohort, and refractory to second line and more were almost 70% in these trials. The tumor volume, which was high, is more than 200 in almost 50% of the patients, and the extranodal disease was present in 50% of the patients. These are heavily treated patients with a bulky disease and represent all sorts of poor biology patients that were taken. You know, Tata being a tertiary care center, and all the referrals comes to them after treatment with all the lines of therapy. So they represent the worst cohort of the patients that can be treated at that point of time. So you see, the LDH was raised in almost 70 to 80% of the patients, and CAR hematotoxic score, which we generally do for assessing the toxicities and other profiles, which was also raised in more than 30% of the patients. If you see, in NHL, 38% of the patients receive some kind of cytoreductive therapies and bridging therapy to stabilize the disease. In ALL, 67% of the patients required some bridging formation. So in the trials, almost 64 patients were enrolled, and 57 patients were infused. Majority of them were BNHL, and BAL were 15 patients. Next slide. So coming to the safety profile, as highlighted, the average duration of hospitalization for NHL and ALL was 10 days. For BNHL, it was 7 days only. And if you see, the ICU admission rates are very low. It is less than 15%, and the duration of ICU admission was almost 3 to 4 days in both NHL and ALL cohort. The CRS rate was 60 to 70% in both the cohort. If you see, the grade 3 and grade 4 CRS, which was absent which was less than 5% in BNHL, and it was around 20% in the BALL group. As you see, these were the refractory patients, have a higher blast percentage in the blood, so they have some CRS. If you compare with the Western counterpart products and some Spanish products and other products, the rate of CRS and ICANS were considerably low, as compared to 20 to 30% versus less than 5% for grade 3 and grade 4 toxicities. Then due to cytoreduction and the lymphodepletion, you have anemia, thrombocytopenia, and neutropenia, which generally recover by day 14. Then ICHS, immune effector cell, lymphocytic syndrome, the toxicity varies from 7 to 14%, but grade 3 and grade 4 was less than 5%. Hypogammaglobulinemia secondary to B-cell aplasia was observed in 50% of the patients requiring immunoglobulin support. So if you see the major toxicities for any CAR-T, the major concerns are CRS and ICANS, and grade 3 and grade 4 was less than 5% versus 20. For CAR-T cell therapy. So not only it reduces the cost, it also gives a comfort to the patient that they have a very less toxicities, and that makes the product safe. Next. So long-term efficacy, if you see, and as we come to the new data, so median survival 12 months in heavily pretreated patients. Otherwise, these patients who have not received at the CAR-T at that point of time, they might be having a survival of at max of 1 to 3 months. So almost the 50% of the patients were able to survive for 12 months and more. And in ALL, the median survival was not reached in those trial patients. Next. So coming to the patient early in the trial, and now in the real world setting, the patient selections has become better. BALL, out of 145 patients who were leukocytes, 118 were evaluated. This is still 31st May 2025, and if you see the results, so one more important thing is, can you go back in the previous slide? So if you see, around 40% of the patients who are leukocytes, they are not able to take CAR-T because their disease relapse, because of their disease progression, and they die due to multiple reasons, maybe infection and many things. Here, if you see, out of 145, death before infusion is only 9, which is less than 10%, as compared to 40% in the Western data. That highlights the quick vein-to-vein time, quick supply of CAR-T cell therapy, and it ensures that maximum patients get CAR-T in quick time. Next. So the outcomes and the results are robust. At day 28, 87% of the patients are in complete remission. These are the refractory patients. And at third month, 75% of the patients remain in complete remission. That shows, means it seems like we are treating ALL patient on frontline BFM or some other protocol when we see these kinds of results. And here the difference is we are seeing these kinds of results in relapse refractory setting and even after post-transplant. So these results are really encouraging. And next slide. So the median PFS and and OS for ALL patients is not reached over a period of 12 months. And this data is also mimicking the Western data of right to cell, where almost 40% patients over a period of 5 years remain in complete response. So for the ALL, if you see the outcome varies with the disease burden. Your progression-free survival depends on the disease burden. Those patients who have a higher blast percentage, their 12 months progression-free survival is almost 40%. These are the patients who are less refractory to even immunotherapy as well. But if you can provide cure to 40% of these kinds of patients who otherwise are not able to survive maybe for weeks, that is great. So overall survival, even in patients with blast percentage more than 5%, is 52%. And those patients who are identified early, in which we are able to reduce blast percentage, the overall survival is almost 80% over 12 months. Next. Next slide. Next slide. So, as we were talking about the trial data for toxicities in the real world also, your grade 3 and grade 4 cytokine release syndrome is less than 10%, and ICANS, which means the dreaded complications, that is for grade 3 and grade 4, it is less than 5 and equivalent to 5%. Hypogammaglobulinemia is 50%. And immune effector cell hemophagocytic syndromes or cytopenias remain 20 to 25%. But these are mostly manageable toxicities. Even grade 3 and grade 4 toxicities, now we have understood how to identify earlier, and I believe in coming few years we will see these numbers even less. Next. So manufacturing characteristics of this product, overall success for the manufacturing was 98% and more than 98%. We have seen patients CAR-T manufacturing even when a blast were 80%, and they have given a good MRD negativity for these patients. So in general, the turnaround period for the for the manufacturing is around 19 days. At max, it varies from 13 to, in majority of the cases, it starts from 14 to 18 days, but the average is 19 days, which is pretty good enough. And vein-to-vein time is also very fast, so I don't think that patient needs to wait too much. And this is highlighted by low death before infusion. Next. So for NHL also, out of 194 patients, 140 were evaluable, and here also the death before infusion was 14 only, and disease progression in 2 patients. Manufacturing failure out of 194 was only 3. So you can see how robust is the manufacturing system for next 19. Next. So in the B NHL cohort, majority of them were male, age 53 in the real world setting, and 50% of the patient have received more than 3 lines of therapy, 8% of the patient have prior transplant, almost 1/4 of the patient had a bulky disease to go with CAR-T infusion. And next. Next slide, please. So, if we see the response, BNHL represents the most heterogeneous cases, and the bulky disease, the biology of bulky disease closely mimics like solid tumor, especially when the tumor burden is too high. So even in those patients, the CR rate is 58%, and it remained like the same at 3rd month. And So these are kind of a patients without any treatment. They have a survival of 2 weeks to few months only. Next. So if you see the patients with a bulky disease, even 9-month PFS, in fact 12-month PFS is almost 45 to 50%, and those patients who are non-bulky, their 1 year PFS has not reached. In overall, in the patients with non-bulky and bulky disease, the median overall survival has not reached yet in the real world setting, as compared to the trial patients when the median overall survival was around 50%. Now you are seeing median overall survival ranging from 60 to 70%. So there is an improving trend in the outcomes of CAR T cell therapies with a better usage of CAR T cell therapy and better training of CAR T. Next. And toxicities in the real world settings, they are pretty low. Grade 3, grade 4, 6%. CRS, ICANS less than 5%, even 3% now. And hypogammaglobulinemia 52%. And ICHS in the trial was 22%, whereas in the real world setting it is 16%. So this trend highlights a better optimization of CAR T cell therapy, better outcomes with the CAR T cell therapy as we move on, as we understand this science, and going forward we will be able to cure more and more patients with CAR T cell therapy who are otherwise palliated. Next. Next slide, please. In ALL, actually in NextCar 19 has set a new standard for care, primarily in the developing countries where your product is much more efficacious than the Western product, and even better in some aspects, especially in the safety part, and at a cost which is less than 1/10 of the Western product. So we are able to produce a kind of product which has a best efficacy and best safety profile, and at a fraction of cost as compared to the Western CAR Ts. So 85% complete response in relapsed refractory ALL. This is not a data for PFM protocol in upfront. This is the data for relapsed refractory ALL, and with 62% overall response in NHL, with progression-free survival, median progression-free survival over 12 months is not reached in both ALL and NHL cohort, and you have more than 500 patients data right now. With this extensive and robust data, we should be grateful, and we are grateful to you for producing this product at an unmatched affordability. I think, can we move to next slide, or Dr. Joydeep sir, you want to take over from here?
Kunal, thank you very much. It's a very good, as usual, a very good representation of the product that is being manufactured by ImmunoACT. So we will continue with our presentation. Now, can we go up on this?
Yeah, I will ask the presentation.
So we will continue with the presentation here. We're just going to move out a little bit. We'll come to back to CAR T again, but we'll just move out a little bit and talk about. It's a little bit funny. This presentation is, but I thought it gives us a philosophical edge Sunday morning to think whether what we are doing is appropriate or not. And The idea of the presentation first came from Peter Gale, Robert Peter Gale. I don't know whether many of you have met him or not, but he is a man who, teaches us to question everything. So that is something which I learned from him. All the, his fruits needed for